Welcome to the Bio Insights, where science comes alive.
If you've ever looked at the elegant, but let's be real, sometimes totally intimidating machinery of biology and wanted to just master it, you are in the exact right place. Today we are demystifying michaelis-menten kinetics. We're going to take what looks like, abstract biological math and translate it into a completely understandable story about microscopic cellular factories. By the time we wrap up, you'll see exactly how these heavy mathematical formulas actually map perfectly onto physical biological realities.
Okay, so let's dive into this. Picture the inside of a living cell. It's not just a blob, it's a bustling, highly efficient microscopic factory. And in this factory, enzymes are the tireless machines literally powering life itself. They pull in raw materials, process them, and just churn out the finished goods our bodies need to survive. But here's the billion-dollar question for scientists. How do we actually measure how fast and how efficiently these tiny machines work? Well, that is exactly what we're unpacking today.
Here's our quick roadmap for the explainer today. We'll start with the enzyme-substrate reaction, then hit the Michaelis-Menten equation, look at 3 simplifying key assumptions, decode the Michaelis constant, and finally, wrap up with reaction orders and saturation.
Kicking things off with Section 1, the enzyme-substrate reaction.
It all starts with a sort of biological meet-cute. Step 1 is reversible binding. Our raw material, which we call the substrate, or just S, bumps into our machine, the enzyme, E. They lock together to form an ES complex. But it's just a temporary union, right? Because from there, we move right into Step 2, irreversible product formation. The enzyme processes that substrate, releases a finished product, P, and bam, the original enzyme is completely freed up to go grab another piece of raw material. So it's basically free enzyme plus substrate forms a complex, which then spits out a product and regenerates the machine to do it all over again.
Moving on to Section 2, the Michaelis-Menten equation.
So we get to the foundational equation. Now, I know math can look pretty intimidating at first glance, but honestly, do not worry. We're going to decode these variables piece by piece until you completely own this concept. At its very core, this whole equation is simply quantifying the relationship between the concentration of our raw material, the substrate, and the initial velocity or speed of our factory's output. That's it.
Now, what's really interesting about all this is how these variables highlight the physical limits of our microscopic machines. Let's break down V0 and Vmax. V0 is just the initial reaction velocity. It basically answers the question, hey, how fast is the factory producing goods right at this exact moment? Vmax, on the other hand, is the absolute maximal velocity. Just imagine every single machine in the entire factory working as fast as physically possible. No downtime. They are completely saturated with raw material. I mean, they can only work so fast, right? So Vmax represents that ultimate unbreakable speed limit.
That brings us to Section 3, 3 simplifying key assumptions. For the math to work, we have rules. The first rule is Assumption 1, excess substrate. This assumes that the concentration of our substrate is much, much greater than the concentration of our enzymes. Going back to our factory analogy, this is like having giant mountains of raw material just sitting there waiting to be processed by a relatively tiny handful of machines. Because of this massive imbalance, only a really small fraction of the total substrate is ever actually bound to an enzyme at any one time.
Our second rule is Assumption 2, which is the steady-state assumption. Think of a perfectly balanced scale. We assume the concentration of the enzyme-substrate complex, those specific machines that are actively holding raw material, remains perfectly constant during the reaction. Basically, the exact rate at which these complexes are forming is perfectly equal to the rate at which they're breaking down to release the product. Synthesis completely equals degradation. The scale stays totally balanced, and let me tell you, that assumption heavily simplifies the math for us.
And finally, we have Assumption 3, initial velocity. When we measure the speed of our factory, we do it immediately after mixing the enzymes and substrates together. Now, why would we do that? Well, because we want to completely ignore any reverse reaction. If too much finished product builds up, some of it might actually, kind of accidentally, bind back to the machine and turn back into raw substrate. We don't want that. By taking our measurements right at the starting line, when the product concentration is basically 0, we keep our focus strictly on the forward factory output.
Next up, Section 4, decoding the Michaelis constant.
So what exactly is Km? Mathematically speaking, the Michaelis constant is defined as the specific substrate concentration required to make the factory run at exactly half of its maximum velocity. It is the halfway point to Vmax. What makes Km so incredibly important and honestly just fascinating is that it's a unique, unchanging signature for every specific enzyme-substrate pair. It doesn't matter if you have 10 machines in the room or a billion, that Km signature stays exactly the same. So the crucial point here is that Km actually measures affinity, like how strongly or how passionately the factory machine likes to grab its raw material. If an enzyme has a small Km, it has a high affinity. It only takes a super low concentration of substrate to get the factory running at half speed, because those machines are just fiercely grabbing onto whatever they can find. Conversely, a large Km means a low affinity. The machines are, frankly, a bit lazier, so you have to dump a massive amount of substrate into the factory just to reach that exact same half saturation point.
Bringing us to our last major concept, Section 5, Reaction Orders and Saturation.
The central hyperbolic curve is basically what anchors our entire mathematical model. Imagine tracing the journey of the factory's output. Along the bottom axis, we're adding more and more substrate, and on the vertical axis, we see the reaction velocity rising. At first, it shoots up super quickly, but then it curves. It starts to level off into this flat horizontal line as it approaches that Vmax ceiling we talked about.
That curve is actually showing us two completely distinct phases of factory operation, which chemists call reaction orders. Down at low concentrations of substrate, we have first-order kinetics. Here, there are plenty of free empty machines. If you add just a little more raw material, the velocity increases linearly. It's highly responsive. But as we keep dumping in substrate, we eventually hit zero-order kinetics. This is the saturation point. Now, every single machine is maxed out. They're all busy. You could literally dump 1,000,000 more tons of raw material onto the factory floor, but the velocity is just going to plateau at a constant Vmax. The speed becomes completely independent of how much extra substrate you add.
All right, let's bring this all together. We've taken an intimidating formula and mapped it onto a really logical system. First, we have the reaction model. An enzyme binds to a substrate, forms a complex, and yields a product. Second, the equation, which perfectly describes that curved hyperbolic relationship between reaction speed and the amount of substrate. Third, we learned that Km means affinity. A low Km means a high affinity, and a high Km means low affinity. And finally, saturation limits. The whole system shifts from a highly responsive first-order state to a totally maxed-out zero-order plateau once the enzymes just fill up.
Now, grounding these abstract math concepts in real-world applications is absolutely crucial. For modern researchers, biologists, and pharmacologists, this foundational model is literally relied upon every single day. Understanding exactly how fast an enzyme works and exactly how strong its affinity is for a specific molecule, that is the fundamental basis for creating chemical interventions and designing life-saving medications.
Which leaves us with a pretty fascinating final thought for you to ponder. If we can map these kinetics so precisely, and if we can design precision drugs to specifically target an enzyme's Km, how could artificially altering its affinity change the future of medicine forever? Just think about the sheer power of that. By mastering the mathematical rules of these microscopic biological factories, we actually hold the potential to cure human diseases at their very root. Thank you so much for joining me